Unless you've been living under a rock, I'm sure you've noticed all the discussions around AI lately. You may have noticed even if you do live under a rock. Even Patrick Starr has a TV. And with all this talk of AI comes with it the talks of AI becoming conscious. You know, like a computer coming to life. And while we aren't talking about AI in this video, I do want to talk about a potential new, different kind of living computer. One that operates using things that are already living. One that operates using DNA. And one that the people working on it are even comparing to quantum computers. It's no secret that DNA is a great way to store data. It's how our bodies store the data to make us. The DNA in each one of our cells stores 800 megs of data, and there are trillions of cells in our bodies, so do the math, it's a lot of data. This potential new system, which was published in Nature Nanotechnology about a month ago, allows for 10 million gigabytes of data to be stored in a DNA system the size of a sugar cube. This is a pretty decent jump from the record in 2016, where researchers were able to fit 200 megs worth of DNA data into a space smaller than the size of a pencil tip. There are plenty of issues with storing data in DNA though, one of them being the fragility of DNA. But the people working on this research seem to have solved that problem. And not only that, this system can do everything that a computer can do. These include a substrate analogous to a tape or hard drive that carries data, the abilities for all or specific portions of these data to be erased, replicated, read, and computed upon, and the ability to execute functions in a relatively continuous and programmable manner. And they were able to do this with the help of a structure called a dendrocolloid, which look like what I'm displaying on the screen now. It's a soft polymer that acts like a scaffold for the DNA, which can be dehydrated for long-term storage and then rehydrated for data retrieval. They start at the microscale, but branch off from each other in a hierarchical way to create a network of nanoscale fibers. This morphology creates a structure with a high surface area, which allows us to deposit DNA among the nanofibrils without sacrificing the data density that makes DNA attractive for long-term storage in the first place. This allows the data to be stored at a very high density and makes it easier to read and write on. They basically got the DNA to sit right on these structures, which kept it from moving around. And they used them because they had a lot of surface area and tons of little branches that the DNA can stick to, which is better than the DNA soup that previous research attempts used. It's like how if you were going to try to write on your Cheerios while they're still floating around in your bowl of milk, it would be super difficult. But if you glued them to something that resembled a dendrocolloid, like a stick, it would suddenly become way easier.
What am I doing, man?
To write the data to the DNA, researchers first converted it to sequences of ACGT nucleic acids, which make up DNA, using computer algorithms. They're then able to read it without destroying it by using RNA. They then read that RNA, which is another big improvement. The system is able to delete and then repeatedly reload files, and even perform computations directly in the system using enzymes. The researchers predict that DNA stored on their polymer nanofibrils would have a half-life of around 6,000 years at fridge temperature of 4°C, and an incredible 2 million years if frozen at -18°C. This is in sharp contrast to my stick, which fell apart after one night in the freezer. And of course, they did face some issues. Since the DNA population distribution isn't uniform, meaning that some strands are produced more frequently than others, it made it difficult or expensive to read the data. The process of reading the data itself could even degrade or change it. Still, even with some loss of data, they were able to keep a very high number of strands and read the data accurately even after 10 transcriptions, with the structure itself even helping to preserve the data. You know, unlike my stick. The way they delete and then rewrite the data is pretty clever too. They basically programmed a natural enzyme called DNase, ACE being the scientific suffix meaning to break down, to destroy specific strands of DNA, and then subsequently rewrite it. And they were able to delete over 60% of the strands that they meant to. So it's not perfect, but it was pretty good from a practical perspective. And I imagine this is something they'd probably want to improve in the future. So great, that's how they store and read the data. But I'm thinking that's probably not what you came here to learn about. You want to hear about that juicy computational stuff, the real living computer. So how were they able to take this storage system and turn it into something actually capable of performing computations? It's a little bit confusing, but I'm going to try my best to explain things simply. So try to stay with me. Essentially, they ordered 1,000 short strands of DNA that contained every possible configuration of 3x3 Sudoku puzzles or chessboards. The chessboards only had knights and bishops, and the Sudoku puzzles only had the numbers 1, 2, and 3. They then programmed the RNA to read the DNA in the same process I described before, resulting in an RNA version of every possible board configuration. Now, this is the fun part. They programmed RNase, an enzyme that breaks down RNA. Remember, ACE means destroy. They programmed the RNase using an algorithm comprised of enzymatic logic operations to destroy the board configurations that were incorrect, leaving only the correct RNA board configurations to be then read by the computer. The algorithm itself is a whole other thing that I'm not going to get into right now. It's on the screen and I'll link it down in the description if you want to read more. Okay, so now you know the basics of how this all works. What does it really mean? Like, are there any practical uses for this technology? Well, as we talked about at the beginning of the video, the system's ability to store and compute data in a very small area and then last for potentially millions of years is very useful. We've seen how technology and computers have been able to advance in a relatively short amount of time, by continuing to find ways to store data in smaller and smaller places. There are, of course, limitations to this technology, like how the dendrocolloids themselves take up space, and how the RNA enzymes in this study would be difficult to scale. But it's still an interesting experiment that provides a proof of concept for a DNA computer somewhere down the line, with future work maybe utilizing Toehold switches or CRISPR technology. One r/science redditor even commented on an article about this topic, saying that maybe we could use this technology to encode data into our own junk genomes. I'm not really sure where they got that idea because it really doesn't have anything to do with this article, but it still sounds cool. But with that, I think I'll end it here. I hope you found this video interesting. I linked all the sources below, so please feel free to read through them yourselves and comment anything that I may have gotten wrong. I'm not a big computer guy, so it's especially possible that I got some of the computer parts at the end wrong. Otherwise, please like and comment how you think this technology might be used in the near future. I'm trying to put out videos that cover the gist of new biotech knowledge more often, so please subscribe because you won't want to miss out. See you next time.